Your cardiovascular system continuously leaks. With every beat of the heart, high-pressure blood is forced through thousands of miles of microscopic vessels, and fluid constantly seeps out through tiny pores between the endothelial cells. This simplified capillary bed shows exactly how much fluid we lose. Every single day, about 20 liters of fluid are pushed out of systemic capillaries and into the interstitial space between your tissue cells. Your venous system manages to reabsorb about 17 of those liters, but 3 distinct liters are left behind. If those 3 liters remain uncollected, the interstitial fluid volume continuously expands. The resulting tissue swelling, known as edema, distorts cellular architecture and disrupts basic physiological exchange. To manage this deficit, the body relies on the lymphatic system. It is an open-ended, parallel circulatory matrix designed specifically to recover this lost fluid and, in the process, use it for constant immune surveillance. Without this secondary network constantly sweeping the tissues, the human body would rapidly face 2 fatal outcomes: severe cardiovascular volume depletion and massive tissue swelling. To understand exactly why this continuous leak occurs, we have to look closely at a single layer of capillary cells and the 2 competing biophysical forces that dictate fluid exchange. This diagram shows the arterial end of a capillary. The large arrows pushing outward represent blood hydrostatic pressure. Because blood arriving from the heart is highly pressurized, it forces water and small solutes out of the vessel and into the surrounding tissue. Working against that outward push is colloid osmotic pressure, or oncotic pressure. Large plasma proteins inside the capillary, primarily albumin, are too big to leave the vessel. They attract water, creating a pulling force that tries to draw fluid back into the blood. Watch what happens as blood travels toward the venous end. The hydrostatic pressure drops due to vascular resistance, shrinking the outward push. The concentration of plasma proteins remains constant, so the inward pull takes over. Fluid begins flowing back into the vessel. Because the initial outward push at the arterial end is significantly stronger than the inward pull at the venous end, the reabsorption math is imperfect. More fluid leaves than returns. This permanent biophysical imbalance guarantees a continuous accumulation of fluid in the tissues, mandating a specialized secondary drainage system to clean up the remainder. Positioned right next to these blood capillaries in the interstitial space are the lymphatic capillaries. The cardiovascular system operates as a closed, continuous loop. Lymphatic capillaries, however, are blind-ended. They begin abruptly in the tissues as open-ended tubes. In this cross-section of a lymphatic capillary, you can see how it handles fluid intake. Its walls are formed by overlapping endothelial cells. When the pressure outside the capillary rises from accumulating fluid, these cells act as one-way mini-valves, swinging inward to let fluid enter. To ensure the vessel doesn't simply crush inward under tissue pressure, collagen-anchoring filaments tether the capillary walls to the surrounding connective tissue, holding the lumen open. Once the interstitial fluid, along with dissolved ions and escaped plasma proteins, enters this network, it is officially classified as lymph. There is one notable exception to the fluid composition. In the small intestine, specialized lymphatic capillaries called lacteals leverage this high permeability to absorb large dietary fats, creating a milky lymph called chyle. The overlapping cells create a mechanical, unidirectional trapdoor. Fluid, heavy proteins, and large molecules can easily fall into the lymphatic system, but once the internal pressure rises, the doors snap shut, preventing any backflow. From the microscopic capillary beds, lymph drains into larger, thin-walled collector vessels. Lymph flow does not have a heart to drive it. To maintain forward momentum, these collector vessels rely entirely on internal one-way valves that prevent gravity from pulling the fluid back down into the tissues. This schematic maps the upward trajectory of lymph. Small networks progressively merge into major labeled pathways known as lymphatic trunks. You have the jugular trunks in the neck, subclavian trunks in the thorax. Deep in the abdomen, the lumbar trunks from the lower limbs and pelvic region converge into a distinct dilated sac called the cisterna chyli. The final return to the bloodstream is heavily asymmetric. The right lymphatic duct drains only the upper right quadrant of the body into the right venous junction. The massive thoracic duct channels everything else, carrying lymph from the majority of the body up to the left venous junction. What starts as a microscopic scattered leak at the cellular level is systematically consolidated through this valved hierarchy, safely delivering 3 liters of recovered fluid straight into the systemic venous supply. But before this fluid is allowed back into the bloodstream, it must pass through an extensive auditing process managed by roughly 600 lymph nodes scattered along the vessels. Look at the internal architecture of a single lymph node. It is highly compartmentalized. Beneath the outer dense capsule lies the subcapsular sinus, followed by the outer cortex, the deeper paracortex, and the central medulla. If a virus or bacterium is swept up from the tissue, it enters the node and hits the first line of defense, macrophages residing in the subcapsular sinus. They trap and destroy many microbes the moment they arrive. Simultaneously, dendritic cells that picked up pathogens in the tissues carry those specific antigen fragments deep into the paracortex. Here, the dendritic cells present the antigens to naive T cells. Once a T cell recognizes a matching antigen, it activates, dividing rapidly to form helper T cells and cytotoxic T cells, capable of hunting infected cells. At the same time, B cells in the outer cortex bind directly to incoming antigens. With the aid of T cells, they multiply, form dense germinal centers, and differentiate into plasma cells that pump custom antibodies into the fluid. The audited lymph, now packed with active antibodies and lymphocytes, exits the node through a single efferent vessel at the hilum. A lymph node acts as a biological choke point. It forces tissue fluid through a dense web of immune cells, ensuring pathogens are intercepted and neutralized long before they can reach the heart and disseminate through the blood. Lymph nodes belong to a much wider anatomical network. The hardware of the lymphatic system is divided into 2 distinct functional categories: primary lymphoid organs and secondary lymphoid organs. Primary organs are the production and maturation facilities. B cells and T cells are born in the bone marrow. B cells mature there, while T cells migrate to the thymus to complete their maturation. Once matured, these naive immune cells deploy to the secondary organs. These are the active battlegrounds, the locations where lymphocytes actually encounter antigens and mount an immune response. The spleen is the largest secondary organ. Unlike lymph nodes, which audit lymph, the spleen monitors the blood directly. It captures blood-borne pathogens and filters out damaged red blood cells. Guarding our most vulnerable entry points are the tonsils. They form a ring of lymphoid tissue in the throat, acting as an immediate immune barrier against anything we breathe in or swallow. Finally, protecting the vast surface area of our internal tracts is MALT, or mucosa-associated lymphoid tissue. These scattered immune clusters form structured defensive nodules directly inside the walls of the gut and the airways. While the lymphatic vessels handle the physical pressure gradients required to keep tissues dry, this distributed organ network ensures localized immune forces are pre-positioned wherever pathogens might enter. The entire lymphatic matrix executes 4 simultaneous physiological mandates: draining extracellular fluid, transporting dietary lipids, clearing cellular debris, and maintaining constant immune surveillance. During a standard physical exam, doctors palpate specific regions of the neck, armpits, or groin to check if the lymph nodes there are enlarged. That swelling is the macroscopic result of intense cellular activity. It indicates that dendritic cells and lymphocytes are actively multiplying inside the node to fight an infection in the specific tissue territory draining into it. The lymphatic network operates in the background, recovering the fluid the cardiovascular system cannot hold. Without this continuous biological failsafe, cardiovascular volume plummets and cellular survival collapses.